and I'll talk about why a non-invasive test for AA exposure is important for research and for healthcare. Okay, so AA occurs naturally in some herbs that are widely used as medicine. So we have herbs that are used in China and East Asia, Europe, Brazil, and India. These are all plants that contain naturally occurring ursodeoxycholic acid. AA emerged as a human health problem in the 1990s. About 100 young women developed end-stage kidney disease. They were at a single weight loss clinic in Europe. And it turned out that in this weight loss clinic, they had first been using a harmless herb called, it's actually called Fangji, and they replaced that with what was thought to be an equivalent herb, but an herb that contained ursodeoxycholic acid, actually a different genus of plant altogether. Later on, many of the kidney failure victims developed upper tract urothelial cancers. That's cancer in the upper part of the tube that the lining of the upper part of the tube that drains the kidney. Okay, so AA covalently binds to adenines, and it results in a fairly high number of adenine to thymine mutations. And then here is that trinucleotide mutational signature or spectrum that Jesse was referring to. Let's see, I'm trying to figure this out. Okay, yeah, so here in this plot, each bar represents the total number of single nucleotide substitutions in a particular trinucleotide context. So the highest bar there represents the number of CAG to CTG mutations across the entire genome. So the higher the bar, the more CAG to CTG mutations. AAG to ATG mutations are there, but not as many. And there are very few mutations from C to A or C to anything. So we know this is the AA signature because we've seen it in multiple independent cell line experiments. We've seen it in patients with AA DNA adducts, and we've seen it in mice with AA-induced liver cancers. I just wanted to emphasize again that we're talking about mutations that are mostly bystanders, not the cancer drivers. So in my talk right now, I'm not going to be talking about cancer drivers at all. I would also say, because I think Arnoud may use this term, that the AA signature is sometimes called SBS 22. Okay, so we know that there's AA in multiple cancer types, so liver, bile duct, kidney, upper tract urothelial, sort of the index cancer, and bladder cancer. Taiwan, it turns out, is a known hotspot for AA exposure, and we looked at liver cancers in Taiwan, and somewhat to our astonishment, we found that 78% of those liver cancers had AA exposure. So in this plot, each of the red bars represents the number of mutations in an AA, the number of somatic AA mutations in an exome sequence. And so AA here is the red, right? Also known as SBS 22. It turns out that liver cancers have been fairly widely sequenced, so we could also ask how extensive is AA exposure in approximately 1,600 liver cancers from around the world. And somewhat still to our surprise, we found that AA is actually quite widespread. AA exposure is quite widespread in Asia. So here's Taiwan, still the hotspot, but a high number of exposed tumors in mainland China, a lot in Southeast Asia, a lot in Korea. I think these are probably Asian patients coming to the Mayo for treatment. Okay, so the point is that AA exposure is widespread in East Asia, and the AA almost certainly causes many liver cancers in the world, and possibly in synergy with hepatitis, as well as urinary tract cancers and kidney failure. So why do we need a non-invasive test for AA exposure? First, it's basically impossible to determine presence and levels of AA exposure based on patient recall or prescription records. And for cancer epidemiology, we need to know, for example, how many people had AA exposure but did not develop kidney failure or an AA cancer, that is, as I said before, who did not develop kidney failure or AA cancer. And for healthcare, it provides a test like this would provide opportunities for secondary prevention by enhanced screening of AA exposed patients. We think this is feasible. We know from other people's research that AA mutagenesis is visible in non-tumorous liver tissue and ureter and bladder urothelium. We also know that there are AA mutations in cell-free DNA in urine. And now Arnoud will talk about the preliminary results of our collaborations with Changgung Memorial Hospital and with TwinStrand.
I cannot share at the moment.
I found the stop. Couldn't find the stop share. Yeah. Okay. Yeah. Yeah, I couldn't find the stop share at first, but I found it. Yeah.
That's okay. Let's see. You can see my screen now.
But you're in the editing view, not the presenter view.
What? Why? How? Oh.
It's probably okay.
Okay, yeah, the presenter opens on the other screen, but it's okay. I'll just use this.
Okay.
Okay, sorry for the delay. So I'm Arnoud, I'm in Steve's lab. And as Steve explained, we want to design a test for non-invasive detection of previous AA exposure. So the hotspot for AA exposure is Taiwan. So we collected a cohort of 12 patients from Taiwan who all had urothelial cancers. So we have 12 patients with bladder, ureter, and kidney cancers. And for some of them, we also collected urine and blood. And just to be clear, for the blood and urine, we isolate DNA from the cell pellet. So we cannot detect any cell-free DNA or stuff like that. Oh yeah, my animations don't work like this, of course. Right, so we started characterizing the cohort by doing whole genome sequencing. So here's two mutation spectra of, in this case, this is a tumor from patient 19 who had a ridiculously high load of uracilic acid mutations. In total, about 160,000 mutations were attributed to uracilic acid. And in the same patients, when we sequence the normal, we actually see the same uracilic acid mutational signature. If we look at the variant allele frequencies of these mutations, we see that the mutations in gray in the tumor are quite a bit higher than the blue mutations in the normal. So that shows that these mutations we detect in the normal are actually very small patches of clonal, of small clones in a normal urothelial tissue. On the right here, you can see the signature assignment on the whole genome sequencing data. Red is uracilic acid. Sorry, I'll just go from the top here. This is the mutation load. So we see that in the tumors, the mutation load is obviously much higher than in the normals. In most of the tumors, we see either uracilic acid or ApoBec. So the SBS2 and SBS13 are ApoBec-associated mutagenesis. And the red stuff is the uracilic acid mutagenesis, which is present in 9 out of these 12 tumors. And we see here in 2 of the normals, so one of them is this one, and the normal of tumor 7 also has very clear uracilic acid associated mutagenesis. Going to the duplex sequencing, which you're probably all much more interested in, right? So we had this, this is for patient 19. This person had a cancer of the renal pelvis. So we did whole genome sequencing. We saw AA, and then when we did duplex sequencing, we saw fairly low AA mutagenesis in the tumor and quite clear AA mutagenesis in the normal tissue, right? This can be explained by the fact that the clonal expansion in the tumor makes for much less unique AA mutations that we can detect using the duplex sequencing. More importantly, when we go for look at the blood and the urine, we see actually quite clear uracilic acid associated mutagenesis in both the blood and the urine, right? So the uracilic acid mutations here are those T2A mutations with this main peak of CTG to CAG. Looking at the complete cohort for the duplex sequencing, we first saw, there were some tumors with a very weird mutational signature, which consist of only cytosine mutations. So we performed HDP, which is an alternative of to NMF, what most people probably have heard of, to extract the underlying mutational processes to in a tumor. So we did HDP to extract this, and then we did mutational signature assignment again. So we see here that this duplex sequencing specific signature is actually much more prevalent in the tumors than in the normals. So we think it's real, but we have not had time yet to look in patient tumors whether we can see the same mutational process. Again, if we then go to the blood and the urine samples, interestingly, we see that all blood and urine samples actually show ursolithic acid associated mutagenesis. Basically, here we can already conclude that our our answer is yes, so we can use duplex sequencing to detect AA mutagenesis in blood and urine. Oh, sorry, that's okay. One thing I want to highlight is that we see some overlaps between genotypes. So we saw especially some mutations that are also detected in the blood are seen in, for example, the tumor or the normal tissue or the urine. And in this patient, for example, there's one variant that's actually seen in all. Similar for this patient, right? So we see some mutations that are unique for the blood, unique for normal, unique for the tumor, but there are also mutations that are shared by all three samples. We of course checked, right? These are not germline mutations. They cannot be explained by contamination of reads from other samples in our cohort. And actually, when we look closer at these mutations in the blood, we see that if we look at mutations that are only found in the blood, so for example, these 308 mutations that are exclusive to the blood of this sample, only about 10 to 15% of them are what we call clonal. So the duplex sequencing found these mutations in more than 2 reads, then we call it clonal, right? But if we look at the mutations that are found in blood as well as any other sample, so for example, these 4 or these 10 mutations, they're extremely likely to be clonal events, so that multiple reads have detected it in the blood. So we actually think that part of the mutagenesis that we detect in the tissues, or in this case, sometimes in the urine also, there are mutations that are in the blood, and we're detecting them in the tissue because there's infiltrating immune cells. In conclusion, we can detect AA mutagenesis non-invasively in blood and urine, which we can then use, of course, try to develop for a clinical test. And we actually see some immune infiltrate signal back in the tissues, which I think is important to note because other people using this technique might also encounter this, right? So there's very small, you could almost call them contaminating cells in your tissues, could actually affect the mutagenesis that you see in the end. So our future plans are to validate this mutagenesis immune cells. So we got some more blood, we will sort them, the immune cell populations, and then validate that mutations are really in the immune cells, in the blood cells and not in circulating tumor cells. And we would also like to assess the sensitivity and specificity of duplex sequencing to detect AA in blood and urine samples. I'd quickly like to thank, of course, the clinical collaborators, which were crucial to this work, to arrange everything with the patient. Of course, Steve in my lab and Bin, and the guys from TwinStrand who helped with this work. And that was it for me.